simulation of physical systems is a generic challenge that is well known across a wide variety of fields. I want to illustrate the simulation challenge to you today with one very specific thing, and then I'll broaden from that. And this specific thing has to do with a well-known task known as nitrogen fixation. And nitrogen fixation is a process that happens in nature that involves taking gaseous nitrogen from the atmosphere and then combining that with protons, typically from water, and then producing as a result of this 2 molecules of ammonia. Of course, in natural systems, in biological systems, this is not usually ammonia, but probably ammonium. And yet this is one of the most important processes to biological systems, including your life and my life, because it is the rate of this nitrogen fixation process that limits the production, for example, of food. And the reason this is such an important and interesting reaction can be illustrated by looking at the reaction diagram of the process. So let this be a reaction coordinate. So progress as the reaction proceeds from the left-hand side to the right-hand side of this equation. And the fact is that the reactants coming in have a certain kind of energy. And let me not be too specific about that. But they have to overcome an enormous activation energy barrier before being able to exit the reaction as a product, the ammonia that we have here. This energy difference here is on the order of 50 kilojoules per mole of the reactants. This energy activation barrier here is much higher, on the order of 400 kilojoules per mole. And so, although it's desirable to be able to fixate the nitrogen and produce ammonia, you have to, at least if you have no other recourse, supply a great deal of energy before you can get this recovery of energy, since this is an endothermic reaction. So, many of you may not know, but we have a lot of nitrogens in our body. Almost all of the 50% of the nitrogens in your body, according to an article in Nature in the early 2000s, comes from a single chemical process. And it's remarkable for me to realize and appreciate that so much of our cells, our bodies, our nitrogen is dependent on this process known as the Haber-Bosch process. Circa 1911, and Fritz Haber and Bosch won Nobel Prizes for their work in this area. This is a process which creates ammonia from nitrogen and steam by cracking it. And so it utilizes a metal catalyst at a very high temperature and pressure, 200 megapascals, and something like 450 degrees Celsius in a very large-scale industrial process to be able to generate ammonia. And what is the ammonia then used for? It turns out that it is used for fertilizer. And that fertilizer then goes into you, the human body. And we can estimate that it probably is responsible for something on the order of the sustenance of maybe 40% of the world population. Enormous industrial scale production. And at the same time, the cost is between 1% and 2% of the world's energy. Okay. Now, this is fascinating. It's a strange place, perhaps, to be talking about and starting a lecture on quantum computation. But here's the idea. Nitrogen fixation is not done by nature using the Haber-Bosch process, using this very high temperature, very high pressure industrial process. Instead, nature does it with catalysts, not metal catalysts, but biological catalysts, which are enzymes, for example, in bacteria, at room temperature and at room pressure. And it does it in a remarkably different way, which we do not understand today. And the example of how this is done is with a special enzyme known as nitrogenase. And this is a bacterial enzyme one of many different kinds of enzymes which are able to fixate nitrogen from the atmosphere and turn it into ammonium and ammonia. But what is remarkable about this enzyme is that we do not understand today how and why it works. Maybe on the order of a dozen other kinds of varieties of biological enzymes have been manufactured or extracted to accomplish the same task that the Haber-Bosch process does. But none of them are quite stable and efficient and long-running as what happens in biology today. Now, the reason we don't understand this is because this is all happening in a complex molecule. The actual reaction, which I wrote over there, involves much more than just nitrogen and two molecules of water or hydrogen coming in to give you let's balance our reaction over here. There are many other things that are happening inside these processes. For example, there are 8 protons and 8 electrons that come in typically. And this may also involve, for example, a proton pump with adenosine triphosphate, losing phosphorus to become on the other side 16 ADP and 16 phosphorus like this. So nature uses a very small amount of energy to be able to catalyze this and take this nitrogen fixation reaction over the activation energy barrier. Now, why is this possible? It turns out there is this amazing center inside this nitrogenase called the iron molybdenum complex. And it has on the order of 100 kilodaltons of atoms inside this. So we're talking on the order of maybe 1,000 carbon atoms. And they're all shaped in various ways with alpha helices and beta sheets. And these shapes then allow the nitrogen and the hydrogen coming in to dock in a certain way. And then because of where they're docked, they can react and do so much more efficiently than they are able to do in this heterogeneous type of catalytic reaction that's industrially done. So we would love to, in quantum chemistry, synthesize molecules which have such functionality, because you could catalyze all sorts of other reactions. We cannot understand why this is happening from a fundamental standpoint today, because we do not know what is happening in a quantum mechanical way with this molecule. Only very recently was even the structure of the molecule understood, like with X-ray crystallography and the like. So we, in principle, could do so. In fact, in principle, we can write down all we need in order to figure out what's happening in this, because the system is actually a very simple Hamiltonian in principle. there's the kinetic energy of the nucleons. There's a kinetic energy of the electrons. And then there are interaction terms between the nucleons and nucleons, between the nucleons and the electrons, and the electrons and the electrons. And I'll expand into this Hamiltonian in a little bit to show you concretely what this means. But suffice to say that we can write down a very accurate description of the system. What we cannot do, what we do not know how to do, is to solve for things like the reaction rate, and even more simply, just the ground state of the system. So at least we would like to know the ground state of the system. We would also like to know the eigenstates. And then we would like to put these all into reaction surfaces, which allow us to tell about what the chemistry of the molecule is going to be. How is it going to react and why? Now, in principle, this is something that a quantum computer can tell us in a way which is fascinatingly different. So as Peter Shor told you about, in 1985, Richard Feynman observed that quantum computers, in principle, are capable of simulating physical systems much more efficiently, faster than classical computers can. Fundamentally, this is because the quantum computer is executing the same dynamics as the quantum system that you're interested in solving and simulating. And then the challenge is to take the quantum dynamics of your quantum computer and map those into the quantum dynamics of the system that you would like to simulate. And so what I want to tell you today is how that is done, what the challenges are, and what the opportunities are for that kind of a simulation.